Good morning, students. I hope I'm audible.
Good morning, ma'am.
You're audible.
Good morning, ma'am.
So, okay, so in plant tissue culture, the last topic that we discussed was about somatic embryogenesis, and today we are going to take up micropropagation. So before we start micropropagation, we'll quickly revise what we did last time. So we discussed about somatic embryos and what is the difference between somatic embryogenesis and zygotic embryogenesis, and what are somatic embryos and what are zygotic embryos, and there are certain differences in the process also. And then the applications of somatic embryogenesis in detail we discussed, right? So quickly, can someone tell what is somatic embryo? Okay, Manaswani.
Ma'am, so a somatic embryo is any embryo which has risen from a cell which is non-zygotic. That is a cell which is a somatic cell. So it's not usually involved in reproduction, and it's used a lot in plant tissue culture.
Okay, okay. Yes, so yes, the next question is what are zygotic embryos? Yes, can anyone tell me about the zygotic embryos? जो मनस्विनी ने कहा उसके बिल्कुल ऑपोजिट इसका आंसर है, राइट?
Ma'am, where the zygote gives rise to the multicellular embryo, it is known as zygotic embryo.
Okay. And zygote are formed through? Yes, Shivani.
Ma'am, double fertilization of ovule.
Okay, fine. So, and what are the applications of somatic embryogenesis? How this can be used in various ways? Namrata?
Ma'am, in organogenesis, then in mutagenic studies, and
Okay. Just stop. Next, Devanshi. Yes, other applications.
Ma'am, conservation of genetic resources, useful for genetic manipulation techniques.
Okay. First, stop. Shivani, others.
Ma'am, genetic transformation.
Genetic transformations. Yes, and yes. Anything else left? So large scale production propagation as compared to zygotic embryo. It is. And yes, it is used in genetic manipulation. It is useful for mutagenic studies. There are so many others, right? एक सबसे कॉमन जो हमें सारी प्लांट टिश्यू कल्चर टेक्निक से मिलता है एक एप्लीकेशन
Production
Ma'am,
of
seeds.
yes
Ma'am,
Ishita?
production of synthetic seed.
Yes
Seed generation.
yes लेकिन एक कॉमन जब हम प्लांट टिश्यू कल्चर एप्लीकेशंस करते हैं सबसे कॉमन एक एप्लीकेशन है जो मैंने उस दिन भी बताया था आपको कि मोस्टली बीइंग यस युक्ता yes Yukta unmute yourself.
Ma'am, I said production of pathogen-free plant. That's the
Yes, that is yes, Yukta, that's right. The production of pathogen-free plant, जो mostly एक it's important technique which is being used for plant tissue culture is being used for this technique. Yes. Okay, so this is and then okay, conservation of genetic resources that you've already said, right? So today we are going to discuss about another technique called a micropropagation. So micropropagation is the basically multiplication using vegetative propagation is one term which is commonly used here, right? So using the vegetative parts of the plant, we are multiplying the plant by this method. And there are a number of methods which come under the micropropagation, right? So if we talk about micropropagation, it is again an artificial process where we produce plants vegetatively through tissue culture or a cell culture technique, right? When we studied in a plant science, when we studied cell reproduction, so there also they were in fungi or in algae or in higher plants, we studied that there were two methods of reproduction, sexual method and asexual method. Asexual methods may vegetative propagation was one method, right? Where we studied that through vegetative propagation, some plants they multiply, such as fragmentation हो गया या आपके ट्यूबर से प्लांट ग्रो हो गए, आपका एलोवेरा प्लांट है, यू सी योर एलोवेरा प्लांट यू आर ग्रोइंग इन द प्लांट पॉट, तो उसी का स्टेम का कोई पार्ट अगर कट करके पॉट में लगाओगे, थ्रू दैट यू कैन ग्रो अ न्यू प्लांट, राइट? रोज में आप कटिंग्स के थ्रू लगा सकते हो. So all these methods are called as a vegetative propagation method, right? So in this artificial process of propagation, plants they are produced in vitro by asexual means of reproduction or vegetative propagation, right? So we know that plants we can produce both sexually and asexually via vegetative parts, or sexually by the seed production also. So one of the means of asexual reproduction is by multiplying genetic replicas of plants that are referred to as a clonal propagation. Clonal propagation means here that since it is an asexual mean of a reproduction, that means no variation is going to be introduced because we are not going for a sexual reproduction here. So clonal means similar kind of a plant. Clonal propagation. We don't want any variation. We want from one plant, 100 of a different plants can be produced. So that is called as a clonal propagation also, right? So wherein plants can be populated from a single individual through asexual mean of a reproduction. So for in vivo propagation, the specific plant's asexual reproduction via multiplication of vegetative part is the only report, since they do not, some plants, they do not generate functional seeds, for example, grapes, banana, etc. So we reproduce, we multiply them by asexual reproduction by using vegetative parts, right? So successful application of a clonal propagation to the following is observed, for example, in case of a potato, in case of an apple, and many ornamental plants. Gerbera ho gaya, aapka Alstroemeria ho gaya. There are many ornamental plants which are being reproduced by this way. So there are different methods of a micropropagation, right? We are just not only, this is not one only common method, but we can use various methods. The first one is the meristem culture. So we all already know about the meristem culture or a meristem tip culture. So this method is the most favorable one when we talk about the production of pathogen-free plants, right? So in this method of a micropropagation, leaf primordial and a meristem is used. Leaf primordia के बीच में जो meristem होता है बहुत small part होता है. It is placed. We use that meristem tip, right? And we put that or place that meristem tip in the media culture and we allow it to grow, right? So after some time, an elongated rooted plantlet, एक proper plant जिसके root भी होंगे, shoot भी होंगे, it will be produced, right? So once after they reach a considerable height, these plantlets are transferred to the soil. And in this method, a disease-free plant can be produced and can be used for a rapid multiplication of a various plants. So in this, we commonly do, I'm not, देखो यहाँ पे just commonly directly बोला है कि plants produce होंगे and then we'll transfer to the field. But last time I told you that hardening is one process in between which is being used. And after that, they are being transferred to the field. Then callous culture. So in this method, a selected plant tissue is placed in artificial growing medium until a callus is formed, right? Callus is an undifferentiated mass of the cells. So after the callus is being produced, they are being transferred to another culture, which has number of plant growth regulators for the shoot and the root production, right? So after few weeks, when a plant is produced, it is being exposed to the environmental conditions, right? So this is again another method. Then we have a suspension culture. So as the name suggests, it is a liquid culture. So in this cells or a group, usually a protoblast culture is being done in a liquid culture medium, right? So in this method of a propagation, cells or a group of cells are dispersed and allowed to grow in aerated and sterile liquid culture media. So this is called a suspension culture, right? Then we have an embryo culture. So in embryo cultures, definitely we use embryo. It is extracted and placed in a culture medium with the proper nutrient in aseptic conditions, right? Then we have protoblast culture. So in this method, the cell is isolated and cultured in appropriate medium to reform the cell walls and callus. So it is a long process. Protoblast culture, we are going to do in a detail when we'll study the somatic hybridization. So how we isolate protoplast and then how we fuse protoplast and all those processes that you're going to study. So this is when we we isolate the plant cells. So we basically isolate the protoplast, right? We remove the cell walls and we expose the protoplast and then we culture it. So that is called a protoblast culture. Later, under suitable conditions, cells they develop cell walls, which again then followed by increase in cell division and cellular differentiation, and it leads to a again new plant. There are various stages in a this micropropagation. So from stage 0 to stage 4. Stage 0 is a selection of explant. Then we go for a culture initiation and establishment, then shoot multiplication, then rooting of the shoots, and then stage 4 is a transfer of plantlet in the greenhouse environment. So stage 0, again the initial stage of a micropropagation. So here stalk plants, right? Stalk plants are those plants, or a mother stalk we call, from which we are using the explant for growing in a tissue culture media, right? So we select those plants that from which plant we are going to use for the multiplication. So the stalk plants are selected and grown under controlled conditions before using them for a culture initiation. So we grow them separately sometimes, and then we use them for a tissue culture processes. Then stage 1, again, we explant are established in a suitable culture medium. So we isolate the explant, we sterilize the explant, we wash them, and then we put them in an appropriate culture medium. So this is again the similar process that in first lecture or second lecture I discussed, that how do we isolate the explant and then we surface sterilize them. We surface sterilize them, or we sterilize them so that there is no contamination. We are talking about aseptic conditions here. We don't want any contamination in our culture. So that's why we take a lot of care at this stage. Then stage 2 is again rapid multiplication of the shoots or rapid somatic embryo formation in a defined culture medium. Then stage 3, shoots are again transferred to the medium for development of the root, where we use the certain hormones for the development of the roots, or the shoots are either transferred directly in the soil for root, or sometimes they can be transferred to the pots also, so that roots can be, the germination of the roots can take place there, right? Then stage 4 is the plantlets are established in the soils. The shoots from the lab are transferred to the greenhouse under controlled conditions of a temperature humidity. So this part is a hardening part, that we don't expose directly them to the environment. We put them in a greenhouse under controlled conditions for some more time, right? So what are the advantages? Again, the micropropagation technique, this is again alternative method for vegetative propagation with enhanced multiplication rate. So naturally also without tissue culture, we are going for a vegetative propagation, but that method takes a lot of time. But by tissue culture, we can multiply number of the plant in a smaller period of a time. Then large quantities of identical plants again can be obtained from a single plant tissue within a very short time period. So this is again important. The shoot multiplication has a very short cycle, and each cycle results in a logarithmic increase in the number of the shoots. So when we do it in a controlled condition, it takes a very less time for the shoot multiplication also by this method as compared to the if we are doing it naturally. Then small sized propagule can be stored and transported easily. If we are doing this multiplication and culturing at one time, then we can transfer it to the another places also. Then here also germplasm. The last time also we discussed that germplasm can be maintained, and here also germplasm stocks can be maintained for several years using this technique. It also helps in this is also the similar as last one. It also helps in production and maintenance of pathogen-free plant varieties. Then in dioecious plant, the seed progeny yield is 50% male and 50% male. Again, this method helps in obtaining desired sex of the plant also. Then millions of plantlets can be maintained in a culture where number of a plant we can maintain. Genetic uniformity of a propagule can be maintained through this technique, clonal propagation. We used in the beginning this word that it is used for a clonal propagation. It is a cost-effective process. New varieties of a species can be propagated by this method. Then a requirement of a less space in a human resources. Again, we don't depend upon the season for the plants, growing plant, because we are growing it in a artificial environment. Then assist in regenerating genetically modified cells after protoplast fusion. So for that method, we can use this. And often produces healthier plants, leading to quicker growth compared to those plants produced by the conventional method, right? So it is a quicker as compared to the conventional method that we are using. Disadvantages, if we talk, we cannot implement this in all the crops, and the plants. Yes, this could be a problem in this could be a problem mostly in all the tissue culture grown plants. So we need to acclimatize them, right? So plants, they find a problem acclimatizing to the natural environment. So this step, the last step, when we are transferring them to the greenhouse and then to the field, that step require a lot of precautions. So this is again one drawback here, that sometimes it can happen. Key points, again, this is a similar plants with a desired characteristic, tissue culture and a planting rate, then stepwise process where large number of the plants are produced from a single explant. So again, these are the similar. So again, micropropagation technique is being used for regenerating forest plants or medicinal plants or other plants which we're thinking are becoming extinct or are less. So this technique can be exploited there. So forests, they account for 29 to 34% of the land area on the earth, and 60% are gymnosperms and 38% are angiosperms, right? Remaining mixed. So while most of the harvested material is used industrially, a significant portion of hardwood is utilized for a fuel on a worldwide basis. We all know that, right? The number that we are exploiting is more as compared to the number that we are replanting. So somewhere it's a disbalance, right? We are creating a disbalance here. So it is generally accepted that forests are being harvested at a faster rate than they are being regenerated, either naturally or artificially. So shortage of wood and wood products has been forecasted. It's a very earlier report that yes, within a period of a time there'll be shortage of plants, right? So that's why conservation of a biodiversity is very important, that we need to conserve them whatever means are available. So in addition, the rapid and disastrous effect of diseases, pests and fires may jeopardize the very existence of certain tree species. So latest fire कहाँ पे हुई थी? Forest fires, who will tell me? Latest forest fire कहाँ हुई थी? Last year तो yes, Manaswani.
Ma'am, there were the fires in Australia in the beginning of 2020.
2020, and before that Amazon forests, they were totally destroyed. Yeah. So many. So the forest fires are also responsible for the destruction of the plants, and burn down or destroys the habitats of so many forest animals or wild animals which are living there, and also the extinction of many forest plants, right? So definitely there are a lot of things. So, but then tissue culture or micropropagation techniques can be exploited to regenerate, right? So there is urgent need for large number of improved and fast-growing trees, and the tree improvement programs are underway, and clonal propagation methods are available to offer limited possibility for achieving this goal, but still they are offering some method. If we can generate the plant by this method, and then they can be planted in the forest or a tree, so we can still reduce that gap. We can still fill that gap or void which is being created. So potential benefits to use clonal planting stock in a reforestation programs have been long recognized, and a lot of increase in gain can be expected from planting selected clonal propagules rather than the selected seed family. So instead of using seed and then growing trees in a plant, it takes a lot of long time. So we can go for a clonal propagation methods also. However, to achieve the maximum possible genetic gain for forest improvement, both for that, both sexual reproduction and asexual or a vegetative multiplication must be used. वरना एक similar, अगर हम will be using only the vegetative ways of a multiplication, so changes or variations, there are less chances that there will be changes. So similar kind of a trees will be produced, right? And see, there is a drawback when a similar kind of or there is a genetic uniformity in a population, there is a major drawback. Can someone tell me what is that major drawback? If there is a uniformity, even if we can see those kind of uniformity are seen in the human population also, there's a drawback, and similarly in the plant population also. So what could be that drawback? Will tell me. अगर genetic uniformity है किसी population में, yes Manas.
Ma'am, so there is, because it's genetic uniformity, there's absence of variation. So in case there's any kind of threat or any kind of disease that attacks that population, then since all of them are similar, all of them can get affected.
Yes,
At least
very
this is
right.
what I think.
Good, very good, Manaswani. So this is the problem. If there's a genetic uniformity, and if, for example, there was an Irish famine, history part still left in the plant pathology, so I'll be discussing there. So there was an Irish famine. Why this Phytophthora infestans, this fungi totally destroyed the potato? Because it was genetically uniform. Just from a few potato tubers, they divided it, they used it. They again, for the next season, they saved from that and again reused it. So they were found everywhere from it. There was no genetic variation in that the potato crop that was being grown there. So when the pathogen attacked, the total crop was destroyed because it was similar, right? कहीं पे भी variability नहीं थी. हो सकता था अगर genetic variation होती, variability होती, some plants would have been resistant, right? They would have survived. So this is one major drawback of a genetic uniformity, that if any pathogen impact or if any pathogen attack will take place, it will totally, there's a possibility that it will totally destroy that crop. So we will not be able to do much about that. So for that, genetic variability is important. It plays an important role here. Yes, good, Manaswani. So sexual reproduction is important for both introducing new genes to prevent inbreeding also, and achieving genetic gain for those characteristics controlled by additive gene effects also. So asexual reproduction allows multiplication of some elite families or individuals in a family, so that exhibit significant gain due to non-additive gene effects. So this what are additive gene effects and non-additive gene effects, that I'm going to discuss this sometime. This is a part of a genetic, basically. I don't know if you've studied this in genetics, but in genetics, these topics are being discussed, right? So the traditional method of a vegetative propagation of forest trees are rooted cuttings or a rooted needle fascicles or a short root or a dwarf spurs for a pine species, and grafting is being used. And in this, the Cryptomeria japonica has been propagated by cuttings for centuries in Japan, as have a populus and salix in Europe since the last century. So this method, they are using it there, right? Then, however, majority of trees propagation by rooted cuttings is in various stages of development and is often characterized by a rapid loss of rooting capacity of the ramette cutting with increasing age of the parent or a plant that we are using. So one of the main aims of vegetative propagation is to multiply trees old enough to have demonstrated their superior characteristics. So we need to select those kind of trees which have all kind of their characteristics or superior characteristics, right? So generally the percent rooting, speed of rooting, root length, number, and survival and growth in and the year of rooting all decline, particularly when the parent plant is more than 10-year-old. So these things, they reduce. So furthermore, in more conifers, rooted branches cutting, they tend to grow continue to grow with a horizontal orientation and bilateral symmetry until after varying period of a time, the terminal meristem changes to radial symmetry and vertical growth. It starts. to take place. The main advantages of using cell culture as a tool in breeding programs and mass clonal production is its potential for enormous multiplication rate. So we can multiply number of plants in a small period of time. Thus, while a rooted cutting can produce a single plant from which several years later further cuttings are available, even the most limited in vitro culture system, that of a resting bud, with today's technique can produce several auxiliary as well as adventitious shoots which can be used for further production of the plant. So this is one major benefit here. So both these type of shoots can be in turn induced to form additional auxiliary or adventitious shoots. The intent of such clonal propagation is to reproduce large quantities of uniform plants of a selected qualities. Furthermore, since woody plants, they tend to be outbreeders, or uniformity can only be granted by the clonal multiplication. So pioneer, there's a lot of work. These just are the example of some people that work on the culture on the cambial tissues of a wood species. It culminated in the formation of adventitious buds in some Ulmus campestris in the earlier 1940s also. And additional success on separate production of shoots and roots in a broad-leaf trees were reported during 1940s. So it was only at the end of that decade that first report of organogenesis in a conifer, that is shoot formation in a callous culture of a Sikia, it appeared. Then first true plantlets for these two groups were obtained later, namely and longleaf pine and intriploid aspen respectively. So these are some research works people have done for forestry plants basically, right? So there was one point in your module somewhere where there was a point that how plant tissue culture can be used to replenish the forestry tree or to increase the production of the forest trees which are of economic importance, right? So asexual multiplication of both hardwood and softwood using tissue culture can be achieved by three approaches: enhancing auxiliary bud breaking, production of adventitious buds, and somatic embryogenesis. So these three techniques can be used. And first two approaches, they lead to plant-led formation by organogenesis through the production of a unipolar shoots, which then must be, rooted in a multi-stage process. So shoots are being produced. After that, we need to produce the roots also. So in contrast, whereas somatic embryogenesis, we have already discussed this, right? They lead to the formation of an embryo through the steps that are often similar to the zygotic embryogenesis. So steps in a somatic embryogenesis and zygotic embryogenesis, they were more or less similar. So the potential for forming large number of plantlets in vitro increases in the above order. But again, there's a difficulty in the production of the plantlet. So when we talk about a plant tissue culture in a forestry, it is not an easy process. There are a lot of things which affect the plant tissue culture when we talk about the forest trees or hard trees or trees with a, because the different kind of a tree, such as for example, if we talk about certain prunus species, they produce a lot of phenolics in the media, which again further hamper their growth in a plant tissue culture. So a lot of other additions are required, or additives are required to be added in a plant tissue culture so that we can reduce those problems, right? So there are number of steps or number of ways it is being improved, but still plant tissue culture is being used to replenish trees or to increase or to enhance the number of trees in a particular area जहाँ पे वो extinct है, या फिर germplasm maintenance में at least they are being used very commonly for trees also. So this technique is being used there, right? So this is all for today's lecture about micropropagation and its uses. And if there's any question, you can ask your questions, please. Okay, so just as I told you yesterday also, since many techniques in a plant tissue culture, they have a similar kind of application. So do a comparative study, right? When you study, when we'll be done with maximum techniques such as We did embryogenesis. Today we did micropropagation. We'll do somatic hybridization also. So then a comparative study can be done, and in that you can rule out which are the similar and which are the just typical benefits or typical application of each technique, or which are the common applications. So in this way, it will be easy for you to remember also, right? Okay, students. So that's all for now. Okay, I'll take attendance and then I'll take class as 12:15 also, plant pathology. I've preponed this class so that you'll be free at 3, because it was very late, 3:15. So I thought since you were not having any class, so we can prepone this class, right? So after that, you'll be free also, I guess. आपकी कोई class नहीं है उसके बाद, right? So let's take attendance for this class. Ayushi Jain.
Present, ma'am.
Sam.
Present, ma'am.
Devanshi.
Present, ma'am.
Ishita.
Present, ma'am.
Kshitij. Manaswani.
Present, ma'am.
Namrita.
Present, ma'am.
Rupal. Nehal.
Present, ma'am.
Sahiba.
Present, ma'am.
Anjana.
Present, ma'am.
Shakti.
Present, ma'am.
Shivani.
Present, ma'am.
Tanvi.
Present, ma'am.
Pridip.
Present, ma'am.
Vivek. Yukta.
Present, ma'am.
Yuvraj. So in okay, Vivek, Yukta, Yuvraj. Fine. So I'll see you at 12:15 for the plant pathology class. Okay, thank you.
Thank you, ma'am.